So today we're going to talk about immunology of viruses. And again, the talk from last year is posted that goes through hepatitis C and influenza in some detail and an example of a model for both of those. The lecture I'm going to do this year is on more on SARS-CoV-2, but I'm going to do it at least at first in the context of a comparison with influenza, and that's in part because there's a much longer literature on modeling in influenza, and there's just a lot more data available on influenza for those of you that are looking to build models. Notably, though, SARS-CoV-2 is generating so much data so quickly and such well-defined cohorts. There's probably higher resolution data for some aspects of SARS-CoV-2 biology than what you would see for influenza, just because of the nature of the technology now that's been thrown at studying the pandemic. Okay, so I'm going to first give you some background on SARS-CoV-2 and coronaviruses. And then we'll discuss how the immune system works to exert viral control, at least as we understand so far. We'll talk about vaccine platforms and the results from the early vaccination efforts. And at the very end, talk a little bit about the emergence of viral variants. So to compare SARS-CoV-2 to flu, if you watched the video last night, but even if you didn't, flu is what we refer to as a negative sense single-stranded RNA genome. And what that means is that the genetic material for flu is encoded on the negative strand of the RNA, meaning it's sort of upside down and backwards in terms of the coding sequence. And so for influenza to make new proteins, it has to take its template strand in the genome and convert it into the reverse complement of the other strand, so A to T, G to C, and convert it from 3' to 5' back to 5' to 3', and that becomes the messenger RNA that makes new influenza proteins. And so it has 3 different polymerases that it makes that help, that it carries in the virion itself to initiate that process, RNA-dependent RNA polymerases to make new message. It also encodes its genome on 8 different segments, meaning that it's easy for influenza to then recombine with other influenza viruses and trade those segments. And that's one of the major ways that influenza evolves and creates new pandemics, is by swapping those different segments. It also has much more error-prone polymerases than coronavirus, and so it mutates at a significantly faster rate as well. And so this has led to the large number of strains that we have for influenza and the sort of constant evolution of it as a virus. While we're going to talk about the variants for SARS-CoV-2, the intrinsic mutation rate in influenza is actually significantly higher than it is for coronavirus. It's a relatively small genome compared to coronavirus, so about 14 kb compared to about 29 kb, and less than half as many proteins for flu. And most of the proteins are all sort of essential. So there are things like the polymerases that are needed to make new message, and packaging for the genome itself, and then structural proteins to form the virus itself. In contrast, coronaviruses, named because of the structure of the spike protein on the virus surface, are the largest RNA virus that infects humans routinely. It's a positive single-stranded RNA genome, so it's already in the right orientation to be directly transcribed and translated. Again, has lots of structural proteins. We're going to talk a lot about the spike, which is this membrane protein analogous to the HA in influenza, and then various other structural proteins. But it has a large number of non-structural proteins. And in virology, when we talk about non-structural proteins, those are usually host modifiers in some way. And we call them non-structural because they're often also non-essential at some level. The virus doesn't absolutely need it to replicate, but it will use it to help make a better environment for it in the host cell. And just a gigantic genome. And this has been a bit of a hurdle to doing some types of research that we can do more easily in influenza, just because you're covering a lot more real estate in SARS-CoV-2. Their replication cycles are somewhat distinct, but the basic principles are the same between almost all viruses, in that, as we talked about yesterday, there's this need for the virus to get in and then get out of the cell and perform its replication as quickly as possible. And so both viruses are taken into to endosomes within the cell that they target through some sort of receptor-mediated endocytosis. And so the receptor for coronavirus, again, is the spike binding to the ACE2 protein on the host cell, whereas for flu, it's much more promiscuous in that the HA protein, this receptor for flu, binds any sialic acid, which is just a modification on lots of different proteins on the cell surface. So they get inside the endosome, that's how they get inside the cell, but then they have to get out of the endosome to start the replication process because they want to access all of that cellular machinery so that they can replicate. So both viruses have a way to fuse back out of the endosome, again, using those surface proteins, either the HA or the spike. They both have a fusion form. And then coronaviruses are just ready to go. They've got their positive sense RNA, so they can just start translating that RNA. They then have a relatively complicated way of making some of their other proteins through the transcription of these subgenomic segments. And so they don't make that whole 32 kb sequence a lot. They make small pieces of it through a relatively complicated process that's really interesting, but that is kind of outside the scope of this course. And then they replicate in the cytoplasm in these really large structures that looks sort of like organelles, very characteristic of coronaviruses. No other viruses sort of make these structures, but it's a way of the virus sort of claiming a lot of host cell resources to efficiently conduct its replication cycle. Flu, in contrast, one unique thing about flu is that it conducts a lot of its replication inside the host cell nucleus. If you remember, we were talking yesterday about the way the innate immune system works is by trying to find patterns of things that are not where they're supposed to be. And so one thought about why flu moves a lot of its activity inside the nucleus is to trick the cell into not realizing that something is wrong by having various strange RNA types inside the nucleus, because you get a lot of strange RNA types inside the nucleus that never appear outside in the cytoplasm in a normal cell. And then both viruses are going to use the host cell machinery for making proteins, get their proteins to the cell surface, package themselves up, and leave. One really critical thing to think about in terms of the biology is that for coronavirus, for SARS-CoV-2 specifically, and for one other human coronavirus, there's a specific receptor, and that's ACE2, which is part of a regulatory system that interacts with your hormonal system in the body. And these cells, ACE2 is on a lot of different cell types. And so the spike protein will bind to ACE2, and that's how it gets inside the cell, versus HA, which binds to any sialic acid. Again, it's on a lot of cellular proteins, but it's not one specific protein. And so the HA will bind to the sialic acid, and that's what it uses to get inside the cell. So both of these proteins have what we call their pre-fusion form and their post-fusion form. The post-fusion form is this sort of trigger that happens to get the contents of the endosome fused outside of the cell, fused outside of the endosome, sorry. And again, both coronavirus and influenza virus do this. And it's this sort of violent triggering event where the HAs or the spike are bound to their target, and then they'll sort of flip themselves around and stab themselves into the membrane and open a pore to let out their contents. And we'll come back to this. It's important to think about these two forms because this pre-fusion form is what's going to be seen by your immune system for the most part while the virus is coming into your body. This post-fusion form is something that's only going to occur inside the endosome, inside the infected cell. And that's true for both HA and for corona. Corona modulates the innate immune response quite a bit through a number of different mechanisms. And so, as I said, it has all of these proteins that are sort of encoded, these tiny little proteins, many of which we don't really have a great function for. There's just been a lot less study of coronavirus biology over the years. The number of labs that focus on corona versus that focus on flu is much smaller. And so a lot of the names for these proteins are, really generic: NSP1, NSP3, 3, 7, it's just the number that they were found in the genome. But it's thought that a lot of the purpose of these proteins is to antagonize the interferon response. And we talked about that yesterday too, as one of the primary ways any cell in the body can mount a response against a virus is by the induction of the cytokine interferon that will induce the interferon response inside the cell that's going to cause all of these non-specific mechanisms to come up that will hopefully shut down viral replication. And coronaviruses are experts, much better than flu really, at shutting down interferon responses in cells that they've infected. There's also a number of other virulence factors encoded by these complex, this complex genome, and many proteins still actually don't have like a fully resolved function. I mentioned that corona has this really interesting way of replicating itself, and so it makes different lengths of its genome. This is pretty common in viruses to have some form of this. When you have a single-stranded virus, HIV does this as well. And the other thing it allows the virus to sort of evolve to do is make different amounts of the different proteins that it needs. And so if you look at each of these subgenomic segments that it's making, each one of these genes gets made every time they're included in one of these segments. And so some of these genes, particularly the N gene here, is made more than almost all of the other genes. And so there was, in a lot of the early testing, the target of the test was for N because it was already known that that was going to be the most abundant. This is a general principle of all coronaviruses, not just SARS-CoV-2. The nomenclature here is you also have these big segments that themselves contain many, many subsegments. And so open reading frame 1A has a huge amount of genes that are important for the replication of the virus. And so subdivided into many, many different protein products. And these will get translated as large polypeptides that get then cleaved into their subsequent constituents. This just shows some of the annotation of the SARS-CoV-2 proteome, sort of structurally. I like this picture. It kind of gives you a sense of the complexity of it. Again, here's the spike complex here that comes off of the S gene, the nucleoprotein complex here, which flu also has a nucleoprotein. This is the protein that helps stabilize and fold the RNA and package it so that it stays inside the virion in a nice tight little compartment. And then the membrane protein that's, again, flu has this too. This is all sort of envelope viruses have, a shell to package everything in, something to package their genomes with, and then a surface receptor that they're going to use to get inside the cell. And then, various proteins that are involved in the replication here, an RNA-directed RNA polymerase, helicase, this is all important for replication. But all these other proteins that you see that are non-structural are probably some kind of host modifier. Coronaviruses also have this proofreading endonuclease. And so this is something, exoribonuclease, this is something that allows it to have a lot more fidelity than a lot of other viruses. And so I said it has a much lower mutation rate despite all the variants than influenza. And that's because it actually encodes actual proofreading activities, sort of like a eukaryotic cell does as well when it's replicating its genome. And again, just to really contrast that with how flu works, where the genome's already in segments, each segment mostly encodes one gene, though there are a few segments that encode multiple genes, usually through the use of an alternate reading frame. And so on the segment 8, there's both NS1 and NEP. On segment 7, there's both M1 and M2. So these are matrix proteins and a ionophore channel. And same thing, structural proteins, HANA surface, the nuclear protein, the matrix proteins. And flu's only really dominant host modifier non-structural protein is this NS1 protein. And the way that NS1 really primarily acts is just by sequestering excess RNA ligands from host cell recognition. It has a number of other assigned activities, but I'd say they're still actually relatively controversial. And the main purpose of NS1 seems to be to sequester RNA. And that's in real contrast to the number of different non-structural proteins that are encoded for by SARS-CoV-2. And so then just summarizing the differences and similarities here, SARS-CoV-2 is a positive sense versus influenza is negative sense, single segment versus multi-segment, very large genome versus a relatively small genome, multiple immune antagonists that are really just going after the ability of the cell to mount this type 1 interferon response that's going to induce all kinds of non-specific antiviral effectors inside the cell and in surrounding cells to try to block the spread of the virus. And sort of a single immune antagonist here, which is just really trying to sequester double-stranded RNA, which is a major inducer of type 1 interferons. And then the specific versus the non-specific receptor. Just want to briefly talk about some of the theories about why SARS-CoV-2 causes disease. I think this is still, sort of surprisingly and fascinatingly not fully resolved. It's even something I'd say we don't understand in all cases of influenza. There's this interplay between the virus-induced damage and the host-induced damage from the immune response itself. That has some sort of feed-forward character in certain individuals. But the idea is in the lung, in particular, when you have a virus that causes a lung disease, that you get infection of the epithelial cells that line the critical air exchange areas in the alveoli here. And so this is what a blown up alveolus looks like. These are epithelial cells here. Can you all still hear me?
Yes.
Okay. My camera or my view shut off. These are epithelial cells that line the alveoli. They're very flat pancake-like cells to allow gas just through their direct damage. You also have some immune cells that live in here, particularly macrophages. And as you start getting that immune response forming, as we talked about yesterday, you start getting more immune cells coming in, and that's going to allow fluid to leak into the alveoli. And so you start getting blockages and reduced gas exchange here. And some of this fluid can be, it's not just like water, it's full of blood components and other proteins and the sort of debris from the immune response, and slowly causing more and more occlusion of the lung. With SARS-CoV-2, there's probably other effects happening systemically that, combined with the reduced capacity of the lung, are causing all kinds of issues. And so trying to understand the character of the immune response and the cells that both play a positive role and a negative role and their relative contributions is something I think that's really open to a mathematical modeling approach, because usually there's not
all your audio keeps breaking up too.
I don't know why. Can you hear me now?
Yeah, it just broke up twice and then it's coming back and it's okay.
Okay. I haven't had any problems this morning. I've been on this link, but maybe the graphics are a little intense. I'll shut off my camera for a second though, just to try to save the bandwidth if that's an issue. Giving me trouble even doing that. Okay. Well, I'm just going to try to get through. If it keeps breaking up, just let me know and I'll try to come back in.
Okay.
Okay. Another really striking feature of SARS-CoV-2 compared to some other infections that have been like flu was that lung damage started occurring really prior to the onset of obvious symptoms, that before someone knew they were sick. This was a really interesting early case report in early stages of the pandemic, where an asymptomatic patient was caught in a routine routine screening from contacts. And they showed this asymptomatic patient, they imaged them, and they already had what they called the ground glass opacities on imaging, which really reflects this alveolar occlusion and the filling up of and blockages in the lung. And this patient only was reporting a mild cough, but had already sustained pretty severe lung damage that they just weren't reporting out through normal symptoms. And so part of this probably actually relates to the fact that SARS-CoV-2 does such a good job of shutting down the interferon response. When you have a robust interferon response and it leads to those early inflammatory cytokines like TNF, IL-1, IL-6, that's what makes you feel sick, and you're aware that you're sick. Here, you can actually sustain quite a bit of lung damage and still be able to breathe without realizing that you're undergoing that kind of damage. So, so then the question is, what's the effective immune response against SARS-CoV-2? And this is going to be a recapitulation a bit from what we've been talking about, but that's a lot, as you'll see throughout the course. It's the same theme sort of over and over again, but with different effector mechanisms usually emphasized depending on the lifestyle of the pathogen that you're targeting. And so because this is a virus that's largely intracellular, there's two forms of effector responses that are going to be most critical. And so those include the antibodies that bind to the virus and prevent it from getting into cells to begin with, that will mechanically block it from getting into an infected cell. And the CD8 T cells, the killer cells that are going to be able to kill a virus-infected cell and shut down that factory of virus production. So there are many other immune components that can help limit infection, but these typically do not have memory. Okay. So antibodies, as again, we've talked about, are made by B cells in response to specific antigens. And so the way that antibodies get made is first through activation of B cells in the lymph node. And so you have infection at some epithelial surface. The antigens then make their way into the lymph node where you have CD4 T cells and B cells living together in a quiescent state. And so they're just sitting there waiting for their antigen to come along. And if you get an antigen that can activate a CD4 T cell, again, that's going to be through formal antigen presentation and activation of the T cell receptor, that CD4 T cell will start migrating towards the area that the B cells live. Simultaneously, if you have an antigen that binds to the antibody that is the B cell receptor, that B cell will get activated and it'll start migrating towards the area where the T cells live. And they'll meet in the middle. And as they're migrating, they're starting to secrete chemokines that are another soluble mediator that create gradients to say, hey, I just saw my antigen, I'm an activated cell, I'm an activated CD4 T cell, I would like to meet a B cell that is also activated. The B cells say, I'm an activated B cell, I'd like to meet a CD4 T cell that's also activated. And that's how they'll find each other out of this vast sea of 99.999% other cells that are not specific at all for what you're looking at. Another important thing to keep in mind about when B cells and CD4 T cells meet is that B cells also are antigen presenting cells. So they will take the antigen that they're binding with their antibody, they'll take it inside their cells, they'll process it, and they can present antigen to CD4 T cells specifically because they have MHC class 2 on them. And so they're going to be able to talk to the CD4 T cell and say, this is the antigen that I see. And if the CD4 T cell says, this is the antigen that I see as well, then they're going to form what's called a germinal center reaction that's going to start licensing that B cell to not just make the antibody as its surface receptor, but to start secreting those antibodies. and using those antibodies as effectors to kill, to go out and search and kill the virus. And so this takes, 5 to 7 days to just get started. And then it will mature over time, and the antibodies will just keep getting better and better and better because the B cells will go through affinity maturation, changing the antibody to better bind the antigen itself. So principles here are that you've got to get virus-specific B cells to get help from CD4 T cells, and you'll start getting antibodies that are secreted, and they'll just mechanically block the binding of, for instance, the spike protein to its surface receptor. In contrast, the CD8 T cells are gonna target infected cells that are processing and presenting antigens on MHC class 1. And so they're gonna also get activated in a lymph node. They also live near the CD4 T cells. They're also gonna wait for signals from CD4 T cells that are activated. So this is all sort of quality control to make sure that you have multiple checks, that there's a reason to activate all these arms of the immune system. And then the CD8 T cells have to leave the lymph node and follow inflammatory chemokine and cytokine gradients to go to the site of infection, where they're going to look for infected cells that have processed and presented viral antigens that they're presenting on their surface that they're specific for. So they'll get primed in the lymph node by a dendritic cell, and then they'll go to the site of infection, and then they will kill those cells. And this is part of why you're going to lose capacity in your lung, because not only is the virus infecting cells and killing them, but the CD8 T cells are killing cells as well. So these are these two major forms of protective immunity, and both of these forms of protective immunity will form memory. So antibody responses target the spike protein, including the receptor binding domain, which is the key part of the protein that's going to make that contact with ACE2. And so there's lots of other structural parts of the protein that if an antibody binds, it might not really interrupt the ability of the spike protein to bind to its receptor. And so we really want antibodies that are specific for that small part of the spike protein that actually binds to the ACE2 receptor. You also make antibodies to parts of the virus that aren't visible to the immune system when the virus is intact and infecting. And so people that have been infected make a lot of antibodies to that nuclear protein. This happens in flu as well. And this is still a huge open question in both fields as to whether or not these antibodies are useful for anything. They're not visible to the immune system here when the virus is intact. And so it's a little bit difficult to understand in what way a virus that's been broken open and has some loose contents hanging around would be useful to have antibodies against those pieces. And then the T cell responses target multiple proteins. So this is the beauty of T cells, is that they aren't looking at an intact virus. They are looking at small pieces of the virus that get processed and presented from an infecting cell. So they can see any part of the virus that's made in any part of its life cycle. And use that to kill the infected cell. Okay, so in the context of an infection, we talked yesterday that you get activation of the innate immune response, and that's going to be really critical for activating the antigen-presenting cells and getting antigen to the lymph node and to activate the adaptive immune response. In the context of a vaccine, we have to add that danger signal. We have to add the pathogen-associated molecular patterns that Charlie Janeway described so many years ago to trick the immune system into thinking that there is an immune response. So the innate immune response has little or no memory, but is necessary for activating the adaptive B and T cell response. And they mimic these danger signals to promote nonspecific inflammation. And so they can be elements of a virus if you're trying to trick the immune system into thinking it's being infected with a virus. And so something like viral RNA or viral DNA can be used in the vaccine itself to trigger the response. Alternatively, you can use various lipid formulations that will look like the lipids that are on a bacteria. There's a lot of research into what adjuvant makes a good combination with a particular target antigen.